Assalamu alaikum. Hello and welcome to third International Conference on Applied Zoology 2020 organized by Applied Zoological Society of Pakistan and Quaid-e-Azam University Islamabad. I am Ayesha Khizar from Fisheries and Aquaculture Department, University of Veterinary and Animal Sciences, Lahore, Pakistan, and my title of presentation is Optimum Dietary Calcium Requirement of Hypophthalmichthys molitrix Fingerlings. First of all, introduction. As we know, mostly the aquatic animals require inorganic elements or minerals. In fishes, the minerals are derived from both ways, either from water and diet. Minerals required by fish are calcium, magnesium, and phosphorus, and the calcium requirement in fishes is affected due to different phosphorus level in the diet, water chemistry, and species difference. In addition, calcium play a beneficial role in skeleton system maintenance, growth performance, and physiological processes such as osmoregulation and blood clotting, while the calcium imbalance play a negative role in organisms' body. If the calcium deficiency causes anorexia, poor reduced growth and feed efficiency, while the excess of calcium relative to phosphorus has an opposing impact in growth and survival of some species. The main objective of this study is to determine the growth performance and feed efficiency of silver carp, to determine the proximate composition and alkaline phosphatase enzyme activity in silver carp, and to find out the mineral status of different tissues such as bones, scales, serum, and whole body. The research work was carried out in Fish Seed Hatchery, Department of Fisheries and Aquaculture, UAS, Shawi Campus, Patoki. The main ingredients of purified diet include casein, gelatin, starch, fish oil, mineral mixture which is calcium free, and vitamin premix, monosodium phosphate, cellulose, choline chloride, and the total percentage is 100. In the in the experimental trial, the basal diet containing calcium lactate as a source of calcium and 5 different experimental diets were formulating containing different calcium levels such as 0, 3, 6, 9 and 12 gram per kg. In diet formulation, all the raw ingredients were mixed and dough was formed by adding 15% of water. After dough formation, pellets were found in meat mixer, and after pellets formation, the pellets were dried and stored in self-sealing bags. In fish husbandry, fingerlings were acclimatized, and after acclimatization, 15 fishes were stocked in each replicate and fed with experimental diet twice a day. The physicochemical parameters were kept constant during the whole experimental trial, and the trial was continued for 90 days. At the end of the trial, growth performance was observed, and from final weight data, the maximum final weight was observed in 6 gram per kg diet, and absolute weight gain was also maximum in 6 gram per kg diet, while after 6 gram per kg diet, the 9 gram per kg diet showed maximum results in terms of final weight and absolute weight gain, while in proximate composition, the 6 gram per kg diet also showed maximum results in terms of moisture percent and crude protein percent, while the crude fat percent and ash percent showed decreased results in 6 gram per kg of diet. After 6 gram per kg diet, the maximum moisture was observed in 9 gram per kg diet, and the fat and ash percent was decreased in this diet. In alkaline phosphatase enzyme activity, it was observed that when we increase the calcium levels in the diet, increase the levels of calcium, the ALP activity also increases in the serum. The mineral from analysis from different body tissues in whole body sample, the calcium and phosphorus concentration was maximum in 9 gram per kg diet. The magnesium and zinc concentration was minimum in this diet. The calcium and phosphorus ratio was maximum in 3 gram per kg of diet, while in from scale samples the calcium and phosphorus concentration was maximum in 12 gram per kg diet, and magnesium and zinc concentration was minimum at this level. The calcium and phosphorus level was maximum from scale samples in 9 gram per kg of diet, while from the bone samples the calcium concentration was maximum in 9 gram per kg diet, and phosphorus concentration was maximum in 12 gram per kg of diet, while magnesium and zinc concentration was minimum in 6 gram per kg of diet. Broken line regression was applied on weight gain percent data to find out the optimum calcium requirement of silver carp. After broken line regression analysis, the optimum calcium requirement was observed 8.11 gram per kg. In conclusion, the growth performance, ALP, and mineralization in different body tissues enhanced when calcium was added in the diet of silver carp. Up to a certain range, the addition of calcium in the diet showed decline results. So the optimum requirement of calcium in diet for silver carp ranges up to 8.11 gram per kg. Above this level, the hypophthalmichthys molitrix showed decline results. So it was suggested, or it is helpful in formulating the nutritionally balanced feed and it is also beneficial for farmers which culture silver carp in intensive, semi-intensive and polyculture systems, because silver carp is a phytoplanktivorous species and most cultivated in polyculture systems. Hope that you understand well, and thank you for your attention.